This is medicine, and this is the human heart. Did you know that drugs affect the rhythm of the human heartbeat? How to see the effect of medications on our heart performance? What is the appropriate evaluation method to test the feasibility of a drug? To answer all these questions, let's look at the following video. In 2005, the International Council of Harmonization, or ICH, implemented a method to assess the potential of drugs to induce cardiac arrhythmias. This method is named Guidelines E14 and S7B. E14 is the clinical evaluation method for the proarrhythmic potential of a drug, and S7B is the non-clinical evaluation method. Both assays assess cardiac toxicity of a drug based on the potential for delayed ventricular repolarizations. These guidelines have been derived from the close correlations between QT interval prolongations and thoracic depoints, or TDP, which is one of the fatal arrhythmia symptoms. TDP is the representative heart rhythm disorder caused by drugs in which ventricles contract more than 200 and 250 BPM, and sometimes evolve into ventricular fibrillation in case. The most common mechanism of pharmacologically occurring QT interval prolongation is blockade of the delayed rectifier current channel, which is also known as the Heart channel. Therefore, as a non-clinical guideline, the S7B evaluates the proarrhythmic risks of drugs, whether delayed ventricular repolarizations and QT interval prolongations, according to the inhibition of the Heart channel through in vitro in vivo method. Indeed, although this method has successfully prevented many proarrhythmic potential drugs from going on the market, it has significant limitations. Some drugs delay ventricular repolarizations and prolong the QT interval, but never lead to the TDP. However, the traditional S7B guideline has regulated this drug because of only QT interval prolongations, even though drugs have high potential to treat disease. Therefore, the limitation of low specificity in this traditional method may have stopped the development of potentially valuable therapeutics by strictly regulating them. For this reason, as a new non-clinical evaluation method, the CiPA project has been suggested by many celebrated institutions worldwide. For a more accurate evaluation of a drug safety, the Comprehensive In Vitro Proarrhythmia Assay, or CiPA, was established in a think tank meeting at the U.S. Food and Drug Administration, or FDA, headquarters in 2013 by the sponsor of biopharmaceuticals, scientists, physicians, and the regulatory authorities involved in developing new molecular entities to evaluate potential proarrhythmic. The CiPA project pathway consists of 4 components to drive major clinical or regulatory analysis of arrhythmogenic potential early in drug discovery, in vitro assessment of drug effect in a multiple ionic currents, in silico computer modeling to predict risk, in vitro effects on human stem cell-derived ventricular cardiomyocytes, and in vivo ECG biomarkers in phase 1 clinical trials. Among these 4 components, we are focusing on the second part of in silico modeling of drug effect in multiple ionic currents. We simulate the study using in vitro patch clamp experimental data. Then, we bootstrap the data to get the sufficient drug experimental data using the Markov chain Monte Carlo algorithm model. Through this process, we obtain 2,000 drug samples that will be used as input in the data in silico simulation stage using the O'Hara-Ruby ventricular cell model optimized from DUTA. This simulation stage calculates the AP shape to obtain the slopes of all waveforms. The in silico simulation results are input for the TDP risk classification process. This classification uses machine learning method. The TDP risk level is classified into 3 levels: high risk, intermediate risk, and low risk. If it is stimulated, the simulation system created will be able to focus on the influence of the selected channel. Through this channel, we can see if the channel can still work. If it doesn't, we can see its effect on the channel. Furthermore, We can also see the impact of drugs on cardiac cells in each channel. With these magnifications, we can see which channels are blocked due to the influence of drugs. Besides seeing the effect of each channel, we can also see if the work of cardiac cells with the electromechanical response and mechanoelectric feedback. To see a 3D heart model, we can scale it up again with the impact of drugs that will occur with electrical conduction in the ventricles, such as whether the heart is beating normally. Through this simulation, we can see whether the arrhythmia will be affected or not after being given a specific dose of medicine.